Next up, we have Dr. Cesar Escalante, and he will be talking about tobacco and tomato ring spot virus.
Okay, thank you, Rebecca. Good morning, everyone, or good afternoon, depending on where you are. So today, this is my second time here participating in this webinar series, and we are looking at two nipoviruses. Those are tobacco ring spot virus and tomato ring spot virus, and we're gonna learn a little bit more about those. Okay, so these are two viruses that kind of share the same history. They're both are the nipoviruses in the same family, in the family Zika, and they are single-stranded RNA viruses, have a polyhedral shape, bipartite viruses, and they are not new viruses. But their impact on emerging cucurbit varieties remain very significant. So they both were characterized in the early 20th century. Tobacco ring spot virus was characterized on tobacco, hence the name, and tomato ring spot virus was characterized on tomatoes. So that's why they have those names. But in the 1950s and 60s, they were confirmed as a major pathogen in cucurbits across the Midwest and the southern United States. And so they share a similar history, like I say, because both originally began classified under the same ring spot umbrella due to their nearly identical symptoms, especially in indicator plants. And I'll talk a little bit about that later. But in the U.S., tomato or tobacco ring spot virus was documented in Wisconsin and Texas cucurbit fields around the late 50s. And tobacco or tomato ring spot virus followed the same trajectory. Around that time, those viruses were characterized. So when we talk about cucurbit host, we are looking at the main cucurbit crops like cucumbers, for example, melons, watermelons, and pumpkins. Those are the ones that are affected by these viruses. And while all are susceptible, squash and pumpkins seems to be more or show more dramatic decline when it comes to an infection of these viruses. And what really makes these viruses particularly difficult to manage is their transmission. So they are both transmitted by the dagger nematode, Syphinema. So this is a nematode. It's a parasitic nematode that it's pretty much present in many fields in the United States, and we will talk more about that when it comes to the transmission part of this presentation. But because of their vector, the dagger nematode it's like I say, it's ubiquitous in many North American soils. So in any field with a history of this nematode is essentially a hot zone for these viruses. So we are just seeing in these cucurbits where these viruses are often found in adjacent fruit orchards. That's very, very common that it will also affect cucurbits if they are present next to each other. So that creates a very complex landscape for management. So that's why we have to make sure when we are planting cucurbits in those fields that they don't have historically presence of these nematodes. So tomato ring spot virus is widely reported in many U.S. states for woody and fruit crops. And like I say, they share many same host. So for example, blueberry or cherries, they are host for this virus. And like I said earlier, both are considered endemic to not only North America, but also in Central America. They are widely distributed. And this map here, I have highlighted some of the states where tomato ring spot virus and tobacco ring spot virus have been reported. That, like I always say, that doesn't mean that they are only present here, but this is basically areas where there is a lot of cucurbit production and that we get most report. I know the cucurbit production in California is huge. I would love to hear from Bill or Bob and see what's the situation with this virus in California. As far as I know, and based on the literature, I think there are not many reports of this virus, but I would love to hear that in the Q&A session and see what's the status on in the Pacific states. Symptoms, well, both viruses, they have very similar symptoms. In the case of tomato or tobacco ring spot virus, the newly infected leaves show this very bright mosaic with some plants showing some stunting, as you can see, and sometimes that subsequent leaves are reduced in size and develop this very dark and green color, as you can see here in this picture. And the very particular type of symptoms, and that therefore the name of ring spots is that they can cause those ring spot on the leaves. And when the leaves turns very dark, those rings are very easy to spot. Sometimes the fruit may develop some prominent bumps, especially that's sometimes in watermelons, that's very visible. And like I said, when the growth is affected, especially when the plants are very young, there is going to be a lot of shortened internodes. The flower and bud proliferation will be more abundant, and there is a reduced fruit set. So that's the main problem there. Sometimes young plants might infected very early may show some terminal bud necrosis and severe stunting. So that's very typical. Some plants also, in some cases, they show some recovery, but don't be fooled with that. So the virus still systemic, and the yield loss is already occurred. So it's a very typical characteristics of some plant viruses that will show this up and down in terms of symptoms. So that's very common also for these two nipoviruses. When it comes to diagnosis, accurate diagnosis is critical. I always say that because ring spot virus or ring spot symptoms can mimic other viruses, like for example, cucumber mosaic virus may cause some ring spot symptoms. Papaya ring spot viruses on cucurbits also cause those ring spots, so it's very easy to confuse. So for rapid in lab or field screening, you can do serological test. Immunostrips is one option. You can get results in almost 30 minutes. You already know the results. ELISA, it's a more in-lab kind of test that you can use. There is ELISA and immunostrips available for both of these viruses. However, if you want to have a more high sensitive result, you have to use RT-PCR. This is an RNA virus, so you have to use RT-PCR. That's basically the gold standard for diagnosis of these viruses that basically targets the cut protein. Most of the tests are based on designing primers from the cut protein. So in some cases, people use bioassays, and so those basically we use baiting plants by using healthy cucumber plants in suspected soils to see if the nematodes transmit the virus to new seedlings. So if you see ring spots on your bait plants within a few weeks, then you have a virulliferous nematode population probably there. So, but also if there is a nematology diagnostic lab in your state, you can try to assess the population and see if you have Dagger nematode in your fields. So virus spread and transmission. So again, I already mentioned this. It's transmitted by Syphinema species. I think there is a very specific species that also it's more, I guess, the major vector of this virus, and that's the Syphinema americanus. Basically, as I have here, the cycle. If you have an infected plant, and then the nematode is going to feed on that infected plant, and in a very short distance movement, it's going to go and feed on that healthy plant, and that healthy plant is going to get infected. But that's just one way the virus can be transmitted. The virus can also be transmitted by secondary spreads, like for example, seed or pollen. There is information that's another type of transmission, mechanical, and also insects. But this is a secondary spread of the virus. The rate for which the virus is transmitted by this means is very low compared to the transmission through the nematode. And then there are some epidemiological factors here. So in the field, really, because this these viruses have a very wide host range, more than 47 families, and so it's a pretty wide host range. So weeds are the main enemy there. So dandelions are one of the hosts in many fields. Those are very common, especially here in the Midwest, and they act as a permanent reservoir for the virus. So the virus survives in the weeds, in the roots basically, and sometimes even in the seeds. And the nematodes move from the weed to your crop, and then that's a good way to spread the virus. So weather also might play an important role in terms of in terms of disease severity, because what happens is that if you have a moist in your soil, that's what we want for our crops, that favors the nematode movement. That's better for the nematode to move. So therefore, you might have more spread of the virus. And then high temperatures might speed up the shock symptoms in the plant. So that's a very common response for viruses. And like I mentioned earlier, there are many hosts, not only weeds, but also other fruit crops that are hosts for these two viruses. And so having fields adjacent to fruit crops, it's also a risk in terms of transmission of the virus. There is also a risk of having mixed infections. It's very common with other cucurbit viruses. And like I mentioned already, there are some viruses that can mask those ring spot symptoms. And sometimes there might be viruses that can cause some synergistic effects, meaning that the severity of that disease is going to be more compared to having just single infections. When it comes to management, I would say management requires here a multi-pronged approach. We have to have several factors. And the reason for that is because in most cases, we talk about resistance being the main barrier for managing viruses. But as far as we know, there is not any commercial resistant cucurbit variety that is available out there. There has been some research to identify some sort of resistance, but these genes have not been able to be added to the commercial varieties that we normally use. So the first thing that we need to manage here is the soil. So if you have a high nematode count in your soils, you must consider maybe fumigation or long-term rotation with plants that or crops that are non-host for this nematode. That's a little bit difficult because, like I said, there is the nematode in this case has also a very broad host range. So we're not only talking about the virus, but also the nematode as a vector here, and also being a direct problem for the crop. So that's a little bit of challenge there. But also sanitation, it's important. Try to use clean equipment, especially when you're talking about tractors. Sometimes it's a little difficult to control that, but be mindful that some fields might be infested with this nematode. So trying to clean those tractors is important when you move from one field to another, because that's a way to move the nematodes in a long distance fashion. So you can move one nematode from one field to another one, and that's where you are moving your vectors there. And then, of course, the third part is I will say weed control. You have to make sure you eliminate broadleaf reservoirs like dandelions, for example. It's important, and other hosts for the virus that then you need to be mindful and eliminate to be able to manage the disease. So I will say these two viruses are very complex, because not only because the way they are and the interaction with the plant, but also because of the vectors. It's transmitted by nematodes, so that means you need to deal with two problems there. Like I said earlier, the nematode is also a direct problem for the crop, so you need to manage that. And on top of that, you have a virus that is very problematic for the plant. With that, I can say that if you have any questions or you want to learn more about our resources, just visit our website, and that it's the contact for all the members of this working group. If you have any questions, specific questions, you can reach out to us. And I want to thank the co-chairs of this working group for letting me be part of it and participate in this webinar series. And here is my contact. So thank you.
All right. Thank you, Dr. Escalante.